Good day. Today we're going to do our simple stain. I'm going to be using a fake bacterial sample so that I can talk to you. This one happens to be a broth, a liquid, but we'll talk about what we would need to do if that sample was a solid. So for example, if you were taking your stock from a slant or a plate. First of all, we always want to make sure we're following that aseptic technique, and we want to protect our stock culture. That's the culture that has our pure bacteria that has been cultured for us. So you'll see I have my staining tray. There is a rack on top of the staining tray. I have some slides here. I have my sample along with my loop, and then I have a water bottle full of DI water. I'm going to use Crystal Violet as the stain for my simple or positive stain, and then I also have blotting paper. One of the other things you may want as you're getting used to working with the microbes is a paper clip, or pardon me, not a paper clip, but a clothespin. So I'll tell you why you might use that in just a minute. Again, if I were working with live bacteria, I would not be talking to you as we discussed in aseptic technique. So I'm going to go ahead and start working with my bacteria. First of all, as always, we are going to want to make sure that we are organized in a way so that we are not crossing over. I'm going to go ahead and flame my loop, sterilize that completely so that entire filament is bright red. Again, think about that stock culture as being one that you want to protect and continue to work with. I'm then going to take that stock culture and, as always, open it with my non-dominant hand. I'm going to protect that stock culture by flaming the lip. I'm going to take a sample of my stock culture and once again flame that lip so that I make sure that that stock culture is completely protected. Now, this is a liquid media that I took that sample from. I don't need to worry about diluting the bacteria. Instead, I am just going to apply that bacterial sample onto my slide. I'm going to use an oval pattern to apply it. Once I'm done applying that, I'm going to go ahead and once again sterilize my loop. Keeping in mind, remember, if this was in a real lab, you would not be talking while you were doing this. We do not want to expose our mouths to any kind of bacteria. And we're going to speed up time here a little bit. You have to wait until the bacterial sample on your slide is completely dry. If you have a lot of fluid that you've taken, that's going to take longer. If you've taken a little bit of fluid, it won't take very long at all. Once it is completely dry, again, we're speeding up time as if it were, you're going to take that slide and you're going to need to do a process called heat fixing, where we're trying to now make sure that the bacteria are adhered well to the slide, that we're stuck to the slide. Now, the reason I want this to air dry first is if you had a lot of fluid here and you're applying it into the heat from the Bunsen burner, you may actually volatilize your sample. In other words, you may get bacteria splashed on your face. And we don't want that. We don't want to breathe it in, etc. So let it air dry. And then we're going to run it through that Bunsen burner flame 3 times. Now, I'm going to use my hand, but if you're uncomfortable with that, you can certainly use that clothespin to hold that slide as well. If you're using the clothespin, just be sure you're not catching the clothespin on fire. If you are using your hand, which is what I prefer to do, then we want to make sure that we are conscious of the fact that that slide will not get red hot, right? It's still going to be clear. So if you were to heat fix with your hand, it's the same idea. You're going through that flame 3 times. You're taking about 1 second per pass. And what that is doing, again, is adhering that bacteria to that slide. Now, the bacteria we're using do not have pigments. They are a clear species of bacteria, and so we are going to be using Crystal Violet as a means to make sure that we can visualize that bacteria easily in our microscope. All of the stains that we use, however, do have alcohol as their carrier, and thus they are flammable. So always we are going to turn our flame off when we get to the point that we are ready to apply the stain. They come in squirt bottles. You're simply going to uncap it, and you're going to apply several drops to the slide. Really, you're trying to cover that area that that bacterial smear was on. Now, again, we're going to speed up time. We're going to say that we have added the Crystal Violet, and it has sat for 1 minute. Once 1 minute is up, we're going to use the edge of the slide, hold it up, let it drain that dye down into our staining tray. And then we're going to very gently rinse that with water. And we want to rinse it with water really hitting above where that smear is, so the water's just running down. Your slide does not need to be completely clear. If you're using your water bottle though and you are applying a lot of force right where that smear was, you are likely to in some ways dislodge those bacterial cells, and that's what we want to avoid. Once that is done, then we come into our bibulous paper or blotting paper. We're just going to open that pack up. We're going to put our slide that now has been rinsed but still will contain dye on it, and we're going to just very gently kind of pat it dry or let it sit for a moment. That's going to remove that extra water. Once we're done, we have a slide that we can now look at with the microscope. You will potentially see some color on here still. That's totally fine. And sometimes you'll feel like you rinsed everything off your slide. Sometimes those are the best slides under the microscope. So they're always worthwhile investigating under that compound light microscope. And that is how you do a simple stain.